Hello learners, welcome back to our channel. Today we are going to learn about DNA extraction. Yes, you all know that this is the process of DNA isolation and purification. You may think of hands-on practical stuff with lab-based equipments regarding this topic, but I promise without any of such arrangement, this video is going to be worthwhile. So let's learn. It is already known that only 24 years old Swiss physician Friedrich Michel isolated a novel type of biological molecule from the nuclei of white blood cell. This scientific finding and the protocol of isolation of nuclein, which is known as DNA nowadays, was published in 1871. However, Michelson and Stahl performed experiments using salt density gradient centrifugation protocol for E. coli DNA extraction in 1958. So you can find that it took more than 50 years to become a routine laboratory procedure for DNA extraction. So in 2020, there were a variety of automated procedures and commercially available kits which are used in our institutions or any kind of scientific research centers. In general, I'm going to highlight the basic steps of DNA extraction, and I am going to avoid the detailed protocols given by renowned scientists which are available for specific samples or specific kits or different kinds of other conditions applied for this kind of extraction procedure. At first, we need to weigh. Suppose 200 milligram sample. The sample part would be leaves, roots, grains. It's like from where I could get the quality kind of DNA. So here please note that the extraction of high quality DNA from medicinal plants is difficult because the presence of large quantities of secondary metabolites, polysaccharides and protein. So this may reduce the quality of DNA during precipitation because they also precipitate with the DNA. Here we will use the liquid nitrogen because it will obtain the good quality of DNA. So it is used extensively. So now we will use the mortar pestle to grind specific amount of sample. While grinding, adding 200 microliter pre-warmed C-tab extraction with 1% beta-mercaptoethanol buffer, which facilitates the separation of polysaccharides. So here C-tab extraction is a very important surfactant which is used only in case of plant DNA extraction. This is needed to remember because in case of blood sample or any kind of animal cell DNA extraction, there will be used SDS or sodium dodecyl sulfate, Triton X100. All these are very popular detergent only in case of animal DNA extraction. You must note this because differences between the plant DNA extraction and the animal DNA extraction is very basic from this point of view. Now collecting the grinded sample in Eppendorf tube and adding more C-tab extraction to make it 600 microliter and placing the sample in water bath for 45 minutes at 65 degrees centigrade with continuous inversion during incubation. Why we need to do this process? Because this process ensures that the enzyme break down histone proteins to uncoil the DNA, and the incubation ensures the complete lysis of the cell. After that, we are going to add the equal volume, that is the 600 microliter of chloroform isoamyl alcohol in the ratio with 24:1 in Eppendorf tube. Chloroform increases the efficiency of phenol for denaturation of the protein lipids. It also allows the proper separation of the organic phase and the aqueous phase. Now we are going to use the centrifuge because we need to centrifuge the sample at 12,000 rpm for 15 minutes at 4 degrees centigrade. In this step, we are going to collect the supernatant in new Eppendorf and repeating the previous steps, that is adding the equal volume of chloroform isopropanol alcohol and centrifuge after that. If required, we are going to repeat these steps. After that, adding equal volume of chilled isopropanol in the collected supernatant and placing the sample at -20 degrees centigrade temperature. The reason behind using the isopropanol is increasing the yield of DNA, and I mentioned about the chilled isopropanol. So this cold temperature causes more and more precipitation of DNA. Next day, it is required to repeat the centrifugation process once more, and then we will discard the supernatant again and adding 500 microliter of 70% ethanol, and again we are going to centrifuge it at 6500 rpm for 5 minutes at 4 degrees centigrade. And again we are discarding the supernatant and letting the pellet air dry at room temperature for 1 hour. Finally, dissolving the pellet in TE buffer or the same amount of deionizing water. Using TE buffer indicates the prohibiting degradation from DNAs. Integrity of DNA now can be checked at 0.8% or 1% agarose gel, and if it is not applied immediately, then the DNA samples can be stored at -20 degrees centigrade until further use. I already mentioned about the differences between plant cell DNA extraction and animal cell DNA extractions. The basic differences between both cells are the presence or absence of cell wall. So in case of plant DNA extraction, the removing of cell wall is needed by using homogenization or adding cellulase. The CTAB, a cationic surfactant, is used in case of plant DNA extraction, while the SDS anionic detergent is used to denature the membrane discarding protein in case of animal DNA extraction. In case of plant cells, there is a presence of secondary metabolites which is not present in animal cells. So the differences between plant and animal DNA lie in the sequence of their DNA bases in the helix. So absence and presence of compounds reflect on DNA base sequences. Also, the genomic DNA of plant is often larger than the animal DNA. So we are concluding here that DNA extraction has evolved for the past 151 years and has developed into a diversity of laboratory techniques. So it is indeed a milestone finding in advanced biological fields. Let's calling it a wrap here, and if you have any kind of suggestions and queries regarding this video, please do not forget to comment. Thank you for your patience.